the SFPM. Thanks. This will be recorded. Welcome everyone to the February seminar series for the Society of Functional Precision Medicine. I'm Tony Latai, president of the society. Before we get to a very exciting talk from Reid and Travera, I would like to share a couple things with the people on this call, and that is, can you guys see? Do I need to swap screens, or are you looking at the right screen?
Tony, we actually see your view, which is the next slide. That's the right view.
Okay, yeah. Great. Well, anyway, I wanted to make, I've hinted at this. If for those of you who are attending the AACR meeting in Atlanta, we are going to be having a reception at the AACR. All the information is right in front of you here on this slide. We will also be sending out emails to people who are on our email list and be posting it on social media. So take a picture or screenshot this or whatever you want and put it in your calendar. Please come and join us. It's going to be an open bar, free food, opportunity for networking. You can meet the board, share your ideas, and just meet other investigators interested in this. And there will be some interesting party favors being handed out too. So you can be the first cool kid on your block with SFPM swag. But this will be the Monday evening of the ACR, and it's just a very quick walk from the convention center, sort of across the street from it. So this is Monday evening that you'll serve as a prologue for the real business, which is functional precision medicine has been given a major symposium at the ACR meeting. And here's the information about that. I'm very excited about the three excellent speakers that we have. I'll be sort of chairing it. These are the three speakers, and I really tried to focus on speakers who have some real translational momentum behind what they're doing. And this is the sort of work that's going to be essential to move our field forward. I would like to urge everyone on this call, if you are watching this talk today, you have an interest in functional precision medicine. For this field to prosper, we need to gain attention. To gain attention, we need to have support at major meetings like this. The ACR keeps track of how many people show up at different symposia. So I encourage everyone at this meeting to if you're academic, get everyone in your lab to attend this symposium. If you work at a company, get everyone who's coming from your company to attend this symposium. It benefits us all to get this room as full as possible. So spread the word and get some butts in the seats there for this thing, because it'll I think really make a difference and help us with momentum as we move forward. Finally, I would just like to thank our sponsors. We've been fortunate enough, we've been engaging in a capital campaign, and we're very grateful for the companies listed here who've chosen to really help with the leadership that's needed in this field. I think this field requires the active participation not only of academics like myself, but also commercial interests. And if we want to get these technologies to patients, I think we have to realize it's going to happen through companies that make a product so good that people and insurers are willing to pay for it. And I'd like to recognize Kiatech, Travera, Xylus, Presage, Excienti, and 2Curex for the generous donations in getting us started and giving us a war chest so we can actually start some planning. I think you'll be hearing more from us. I think we're becoming a better functioning society. And as always, we are very happy to hear your input on what we could be doing different or what we could be doing better. With that, I'm going to turn this over to Keith Ligon, our secretary, who will introduce our speaker for today. And let me see if I can stop my share.
Yeah, thanks, Tony. It's a real pleasure to introduce Reid, who's the founding CEO of Travera, precision medicine company. And previously, Cliff was the founding chairman, president, and CEO of Complete Genomics, a leading developer of human whole genome sequencing technologies and services. So he's familiar with getting information and data back to patients. This background really gives him a unique and valuable perspective, I think, to share with us on the FPM field. Prior to his multiple entrepreneurial successes, he had earned an SB in physics from MIT, an MBA from Harvard Business School, and a PhD from Management Science and Engineering at Stanford. Currently, he's on the visiting committee of Biological Engineering Department at MIT, a member of the MIT Corporation Development Committee and advisor to Warburg Pincus. So please do join us in thanking Cliff for sharing his progress and Travera's with us in the community here.
Great, thank you, Tony. Thank you. I'm sorry, thank you. It wasn't Tony at all. Thank you, Keith, and thank you, Tony, for the introduction. And let me just check. Can you guys see my slides?
Yeah, looks good, Cliff.
Great, thank you. All right. So thanks much. So today I'm going to do a kind of a very brief introduction of Travera and then walk you through what we're doing kind of on the product and technology side. I am not the technologist in Travera, I'm the commercialization guy. So I'm going to present this in sort of a commercial context. But I do have joining me Rob Kimmerling. Rob is our Chief Technology Officer, was instrumental along with Mark Stevens and Salim Okum in developing this core technology in the Manalis lab at MIT. And a comment about other people involved. First of all, Keith Ligon, who very graciously introduced me, is actually one of our founders. So for kind of full disclosure there, as is Scott Manalis. Scott is a professor at MIT. And is really the leader who enabled the creation of this core technology that enables us to make Travera a real company. Scott and I and the team launched Travera in 2018 as a spin out of MIT, venture financed it, and I'll kind of describe where we are in the process of commercialization. Okay, so I want to start by describing, again, a little bit from a commercial perspective, the problem that we're setting out to solve here. So what you see here is effectively the last page in the NCCN guidelines for stage 4 breast cancer. So this is recurrent unresectable breast cancer. And so these are, these are metastatic breast cancer patients, and this is no small matter, right? About 100,000 Americans are metastatic breast cancer patients. About 40,000 of them die every year. The other 60,000 would die of metastatic breast cancer, except they die from something else first. So this is not rare at all. And for those 100,000 patients who get to the metastatic stage of breast cancer, this is what they're faced with. If you look under first line here, you see trastuzumab, that's Herceptin, plus a couple of chemos. And then second line, you see Herceptin plus a couple of HER2 inhibitors. And then you get to this very unfortunate part of this page. And what it says is third line and beyond, optimal sequence is not known. All right, well, what you see there, third line and beyond, is 8 combinations of 12 therapies. And this basically is kind of thrown over the wall to the oncologist, and the official position of the NCCN guidelines is we don't know what to do. All right, so I question that as valuable therapy guidance. And I like to say, if you look at the NCCN guidelines for non-small cell lung cancer, it's worse, because this page for non-small cell lung cancer has 27 drug combinations of 13 unique therapies, and they're listed in alphabetical order. Okay, so again, that's not the best therapy guidance you could imagine. And this page exists for all 61 of the NCCN guidelines cancers. So this commercially, we think we can help all cancer patients, but the cancer patients we're focused on are these late stage cancer patients who are simply out of guidance. NCCN guidelines is providing them with virtually nothing of value, and their oncologists are just in pure trial and error mode, mostly just applying lots of different chemos, and most of these patients end up in hospice and death. Okay, so that's the problem we're setting out to solve. So many of you have seen Scott's technology. This is his mass measurement technology, and I'm going to do a very short version of it for those of you who haven't seen it. But there's a lot of depth here and a lot of publications here. But let me give you kind of a little bit of a hand-waving description of what this fundamentally does. So Scott and his team in the Koch Institute at MIT invented a new measurement tool. And as I love to say, measurement tools change science, and this is a radically new measurement tool. And what this measurement tool does is it weighs single cells. All right, it actually can weigh other small particles like protein aggregates, but what we're using it for is to weigh single cells. And what you see on a picture there at the tip of a finger is a picture of the core technology. This is a MEMS chip. It's manufactured in a semiconductor fab right now in Grenoble, France. And it's a fluidics device. You can pump liquids through this MEMS chip, and in particular, pump liquids with single cell suspensions through this chip. And what this chip consists of, and you see kind of an artist rendition there, and here's where I'll do my hand waving physics for you. It's an oscillating cantilever, fancy word for a diving board, right? Inside that oscillating cantilever is a 30 micron diameter fluidics channel that runs down the cantilever and back up the cantilever and connected to input-output ports of the chip. Outside the oscillating cantilever is vacuum to allow it to freely oscillate. And what we do is we take advantage of a very simple law of physics that says that the resonant frequency of an oscillating cantilever is directly proportional to its mass. So when you pump single cells down and back this oscillating cantilever, the resonant frequency changes, and it changes in proportion to the mass of the cell. Now, frequency changes are one of the most accurate measurements you can make in all of physics. You measure that with a $100 FM circuit to an accuracy of about 10 parts in a billion, which gives us this extraordinarily accurate measurement of the mass of the cell. And there's some measurements there in the bottom of the screen, but the one I think is physically the most meaningful is we can detect the change in the diameter of a cell of about 5 nanometers. Okay, wavelength of visible light, 400 to 700 nanometers. So if you're using a microscope to try to measure the cell mass, you can't get anywhere near this kind of accuracy. So a breakthrough measurement tool. No one's ever seen cell masses with this degree of precision ever before, okay? And Scott and his team used it for all sorts of cool cell biology work. But fortunately, Scott hooked up with Keith Ligon and David Weinstock and a few others at Dana-Farber over the years and said, hey, here's an interesting proposition. Can we use it for ex vivo cancer drug effectiveness testing? And as many of you know, that has been a failed field for many, many years now, starting with Oncotech and then Precision Therapeutics and then Notable Labs and then Mitra and Allcyte and all the companies that have tried to do this and, succeeded in AML and failed everywhere else. Said, could we use this for an ex vivo cancer drug effectiveness test? Because it's so sensitive, we might be able to do this really fast. And the really fast comment comes out of the observation that, very simple observation that dead cells weigh a lot less than live cells. But cancer drugs in a lab typically take days, as long as a week, to kill cancer cells. And in our hands, and I think in the hands of a lot of your labs, when you take live cancer cells out of a patient, live solid tumor cells out of a patient, blood cancers are very different. Take live solid tumor cells out of a patient, they die quickly. You have to put them through heroic acts to keep them alive. But if you just put them in minimal media, they're dead in 2 days. And yet the drug is going to kill them in 5, 6, 7 days. So there's a terrible timing problem in ex vivo cancer drug effectiveness testing that's been solved through cell lines and organoids. It's been solved heretofore by growing the cells, keeping them alive long enough. And there's a lot of literature that says, hey, if you pump these cells full of cytokines and growth factors and all the things it takes to keep them alive, they change. Bill Hahn just published a great paper in pancreatic cancer about passaging cells, and they drift phenotypically, they drift genotypically. And that's fine for a drug development assay, not so good for patient testing. So we use the observation that, hey, these cells are going to be a lot smaller when they're dead. And the question is, if we can detect just a few % of mass change, what effectively we're testing is the beginning of cell death, not the end of cell death. And we can do that very quickly. And the question that DFCI and MIT answered, can you do that in 24 hours? The answer is yes, you can. So here's what we do. We take live cancer cells out of a patient, and I'll do the one drug example. We typically run panels of 20 drugs, but conceptually, here's how it works for a single drug. Take 10,000 live cancer cells out of a patient, split them in half, 5,000 cells in each half, put them in 2 different wells of a plate. Add drug to one well, don't add drug to the other well, keep all other conditions identical, incubate for 24 hours, and then weigh each individual cell in each of those 2 wells. Don't make a bulk weight measurement of the wells. Weigh each individual cell in each of those 2 wells. And what you get is a histogram that you see there in the bottom of the slide. You get a weight distribution, bell-shaped curve, because these cells are in all different stages of the cell cycle, so the weights are pretty broad. But what we do is then compare those 2 weight distributions. And it turns out if those 2 weight distributions are statistically identical, then the drug isn't doing anything, at least not in the first 24 hours of exposure to the cells, right? And we say, hey, that drug, it isn't changing the masses even by a few % in the first 24 hours. We believe it's not going to change it by 50% in a week. But if you see those 2 mass distributions shifted from each other in a statistically significant amount, which has always been too small to measure prior to the invention of this measurement tool, then we say, aha, we see the beginning of the activity of this drug, and we say, gee, in 5 days or 7 days or whatever it is, we expect this drug to kill those cells. Okay, That's our biomarker. So this is a mass change biomarker, a completely biophysical biomarker. Has nothing to do with the molecular mechanism of action of the drug. It has to do with how the drugs change the mass of the cell, right? Okay, so I'll talk a little more about that, the core technology and all, but the first question is, okay, you've got a 24-hour measurement of mass change. Does it predict patient outcomes? And the answer to that is, in our limited data set, it seems to be very good at that. So here's our again, very limited data set around actual patient outcomes. And the basic experiment design under which all this data is generated is this. Take live cancer cells out of a cancer patient, get them to us within 24 hours because these cells die really fast. So they're FedEx to us in cold shipping packs in 24 hours. We take the cells out of the pack. We then run a panel of drugs against those cells, okay? And if we know, tissue of origin, we know the patient, we know their pretreatment, we can make a really good guess of what the next therapy is going to be for these patients. And we can run 20 drugs by these patient cells. So we run our panel of drugs. The oncologist then gives the patient whatever drug they choose to give. Chances are they're giving them one of the drugs we tested, okay? Months later, in the blood cancers, it tends to be 1 or 2 months. In the solid tumors, maybe 4 or 5 months. We get back the clinical outcome from the patient. And then we compare the outcome of the patient from our prediction of whether that drug would or would not help that patient or work for that patient. And then we just calculate our predictive accuracy. So as you see here, bottom right, of the 49 patients we've run so far, we've gotten 41 of them right. And then you look into the chart, you realize, roughly speaking, half of those are positive predictions. We said the drug would work and it did. Half are negative predictions. We said the drug wouldn't work and it didn't. We ran about a third, myeloma, AML, and then the last third we ran are solid tumors. Now you'll see there are only 17 solid tumor samples here, so we're not deluded into thinking we've proven a lot here. But the early indications are terrific. We're seeing the responses that we expect to see in the solid tumors. Since putting this data set together, we've really shifted all of our weight to solid tumors now. And over the course of 2023, we expect to add to this data set about 100 patients in solid tumors with positive and negative prediction values. Now, the solid tumors, that covers a lot of ground, right? These are like all the carcinomas. I think 7 of these 17 are non-small cell lung cancer. We got 7 out of those 7 right. But the rest are breast and panc and colon and ovarian and liver and all those kinds of things. And these are late stage patients. A lot of our tests are liver mets. So the tissue of origin is what we report as the cancer, but it's a liver met that we end up testing maybe half of the time here, okay? So it's an early data set, but it's extremely promising, and sort of job one of Travera is expanding this data set. I'm gonna leave one other comment before I move on, and that is, if these kind of predictive accuracy holds, then there's an extraordinary interpretation of this test, and that is that our test result ends up being a strong proxy for the patient's clinical outcome. That has enormous implications for drug developers. In particular, if this were true across lots of drugs and lots of cancers, and I'll show you, we think it is, then in the long run, before a drug developer spends $100 million to run a phase 3 clinical trial, maybe they should take 50 patients and run their investigational compound through our test at $1,000 each or a few $1,000 each. And if they see virtually no response to the compound, don't run the $100 million clinical trial. So this could be a proxy. It's kind of a surrogate clinical trial, not perfect, but a surrogate clinical trial that could potentially save a lot of money for drug development. So some interesting future developments are there, and I'll end this talk on other thoughts about the future. But primarily what we're developing this for is patient testing, and primarily for late stage cancer patients who are out of options under the NCCN guidelines. Okay, and where did this data come from? I just want to point this out. We're running a whole bunch of clinical studies. There's 7 clinical studies on the slide. The first 4, the myeloma study and the first 3 of our solid tumor studies, were all launched before COVID, and COVID pretty much shut them all down. And I don't know if you've ever worked with a clinical study that's been shut down, but it's really hard to regain momentum in a clinical study that's been shut down. And we're really struggling with that. But fortunately, we launched 3 additional clinical studies, the bottom 3 here, Sequoia, NCI, and XCures, during COVID. So they weren't kind of shocked by COVID and the whole restructure of the US clinical trials infrastructure that went on during COVID. So we have 3 clinical studies that are vibrant and moving forward, even though the first 4 are really kind of stalled, and we're working hard to keep them going. But good clinical partners, good clinical data. All right, so there are two key questions we typically get asked. I'm gonna make this reasonably quick talk so you can open it up for questions, and I have Rob to answer your detailed technical questions. But the two questions we get asked a lot are, first of all, how does it work? I mean, okay, mass as a biomarker and mass change in response to cancer drugs, no one's really investigated that in any detail. You know, what are the mass balance equations? How does it, what's physically going on inside of these cancer cells? And part of our answer is, you know, testing 273 anti-cancer agents against 61 different tissues of origin. That's a tall order for a small company to validate all of that. But we've done a bunch of literature searching and have come up with pretty good explanation. Has actually published a lot of this raw data in a publication I'll refer to on the next slide in Communications Biology. But we have a pretty good idea of what's going on for at least the major categories of cancer drugs. So, and the point here is that when we see these different categories, cell cycle arrest, metabolic disruption, structural damage, we see a characteristic mass response, okay? We see kind of a signature in mass response space of each of these different mechanisms of action. And I've been speaking as if cells shrink on their way to death. Turns out for a lot of mechanisms of action, the cells don't shrink at all, they grow. And in particular, you start with the cell cycle arrest drugs. If you have a cell cycle arrest drug that arrests in G0, yeah, those cells shrink, you know, they divide and then they can't grow up. So the whole distribution of masses as the cells divide, it gets smaller. But if you have a drug that arrests in G2 just prior to cell division, well, those, you know, that mass of drugs, the mass of cells over that 24-hour period is going to get slightly bigger as the biggest cells can't divide. So we actually see that signature for the different cell cycle arrest drugs. For the metabolic disruption, most of those just inhibit protein synthesis, but some of them don't. For example, the proteasome inhibitors in myeloma, they actually prevent broken protein reclamation in the cell. So if you use a proteasome inhibitor, you actually see those cells get bigger as they can't reclaim their broken proteins. And then in the structural damage, these are a lot of the actions of the chemos where they just blow up cell membranes. Those tend to move in one direction. You know, those are killing cells, and you end up with this little bimodal distribution, a very characteristic mass response of cancer cells to cancer drugs. So we see these signatures, and we've published these signatures in paper to the point where if you could hand us a drug and not tell us what it is, and hand us a cell line that the drug is going to be sensitive to, we can run the mass response of that drug on that cell line and probably tell you what the mechanism of action is. These are very, very clear signatures in mass response based. And I like to say we just scratched the surface of understanding this biophysical biomarker. It's a brand new measurement tool, and there have been limited numbers of experiments run. So there are a lot of questions we can't answer about how mass response maps to molecular mechanisms of action. But these general kinds of concepts we're pretty comfortable with. The second question I want to ask is, what's your dosage? People always say timing and dosing studies. Okay, you are dosing cells for 24 hours in your lab. What's the dosage? And I'll say we just published a paper. This is in the December issue of Communications Biology in 2022. And this is one of the figures out of the paper. And what it shows is what we've done is essentially run IC50s for all of the drugs. This is 60 plus FDA approved drugs across 12 different cell lines, and then sort of correlated the IC50 with the mass measurement. And the general result I'll cite, although I'll turn this over to Rob at the end to tell you through any more details you'd like, is that kind of the therapeutic range of these drugs for seeing some mass response in a 24-hour period is pretty broad. That is, you run the IC50 and you find that the concentration where you get no response, you run it where you get general toxicity. And the concentration typically has a few orders of magnitude between those two endpoints. And we pick a concentration pretty much in the middle. And it's turned out that for the patient testing that we've done, and we've run now, last year we ran 166 patient samples. So we're beginning to get some decent data sets on patient samples. It looks like there's a pretty big therapeutic window there. And that's, very fortunate for us, because I don't think we have the ability to exactly tweak and tune, the perfect dosage for every drug, but the window seems to be pretty broad, and we have a sort of a common threshold and a common dosage that we use. So we're able to make this work across a pretty broad range of drugs and a pretty raw, in this case, a pretty broad range of cell lines. I'll go on to say that since publishing this paper, since submitting this paper back in the middle of 2022, we've gotten CLIA certification for this whole thing. I've done a lot more validation and kind of a site chart. I'm not going to read you all of this, but what you see across the top of this slide is all of the cancers that we're CLIA certified for, and this is all of the carcinomas, and then includes myeloma, and actually listed here leukemias, although we're finishing our leukemia certification this month. And kind of the breadth of this is a little, breathtaking for such a small company, but it's because to us, a carcinoma is a carcinoma, right? We don't really care much about tissue origin, breast and lung and colon and panc, a little bit around dissociation. But the fundamental cellular mechanism of these cancers responding to cancer drugs seems to be very similar across all the carcinomas. Now, I will say, if you kind of look at in that footprint, the footnote, we don't do the lymphomas. The association there is different. We don't do the sarcomas right now because it's really hard to get live cancer cells out of the sarcoma. And we don't even do bone mets. You probably know a lot of prostate cancers metastasize to bone. And unfortunately, we're not able to help those patients. So we can't get enough cells out of the bone mets. We're not doing the glias yet. That's something we'll do someday. And we've never run a pediatric patient through any of this testing. So we're not doing the pediatric cancers. These will all come in the future. I don't think they're fundamental reasons we can't do it, except maybe the sarcomas. We can never get enough cells. But we're CLIA certified for, this represents about 85% of all the cancers in the United States. We're also CLIA certified now for a pretty broad range of drugs, over 100 FDA approved cancer drugs, most of the popular ones. So you see the inhibitors, the cytotoxics. We, obviously started in multiple myeloma and AML, so we've got a lot of those drugs. What we don't have on here are the antibodies. We've actually done a study of daratumumab and made that work, but it's a complicated assay, and we haven't reduced it to practice yet in our CLIA lab. But I'll direct your attention to the far right column here. The bottom right of the column shows slow-acting drugs we do not test as monotherapy. So it turns out for these 4 drugs, so 5-FU, len and pom in the myeloma space, and pemetrexed, we just don't see enough signal in 24 hours to actually test these drugs. Now, we potentially could do it in 48 or 72 hours, but the cells are dying so fast, it still makes for an unreliable test. The bad news is it doesn't work on all drugs. The good news is it's a very small set of drugs that we've had to disqualify based on the kinetics of the drug. We're actually in the future likely to expand this list of 4 to a list of 5, because gilteritinib in our hands for AML doesn't work very well. We miss as many as we hit, and it just says, hey, we need to do an assay validation of gilteritinib that we haven't done yet. And I spent some time with the notable guys. I want to say thank you to them, Matt DeSilva, who's now moved on, but his team, asking them about gilteritinib, and they said, yeah, it's hell. Gilteritinib is a really hard drug, and we just, given our breadth of footprint, we just don't have time to spend optimizing gilt, but we'll do that, I suspect, next year at some point. Still, the message is this works for a very broad swath of drugs. The other thing I want to direct your attention to here is the immunotherapies. Again, I don't have slides on this. I didn't want to spend the time going through all of that, but what you see is that we're testing, atezo, dostarnivo, and pembro. These are the PD-1, PD-L1 inhibitors. And this is a very different assay that we run. It is CLIA certified. We actually return results of running these drugs when we can to oncologists who order the test. But what it requires is T cells. So we're not measuring the cancer cell response, the mass response of the cancer cells to these drugs. We're measuring the mass response of the T cells to these drugs, with the notion that if the drugs enable T cells to be activated, those T cells grow. They increase their mass. And we can make that measurement, and we clearly see differential response among these drugs in different patient samples. But we have, I think, a total of 3 clinical outcomes so far. So we're long way away from any kind of a clinical validation. That's one of our objectives for this year, is to run a lot of patients through these drugs and figure out if our measurement of T cell response to these drugs is correlated to the patient's outcome. I will say that the only way that practically speaking, we've been able to run these drugs is not when we're getting tissue samples from, fine needle aspirates or core needle biopsies or even surgical resections. It's when we're getting malignant fluids. So we get pleural effusions of the chest cavity, we get ascites of the abdominal cavity, and we get 150 mils of that liquid in 350 ml tubes. And when we do that, you often get, many millions of cells, and you hopefully get a few 100,000 cancer cells, but you often get millions of T cells. So when we get those millions of T cells, then it's very easy for us to run these checkpoint inhibitors against it. So again, sort of a novel use of a new biomarker, a mass-based biomarker, to see if we can come up with a biomarker, maybe an adjunct biomarker to PD-L1 for the checkpoint inhibitors. So we have gone down the path of getting CLIA certification for all of this technology. And part of the reason for that is that our clinical studies, due to COVID, went really, really slowly. We were terribly disappointed our lack of ability to generate clinical outcomes data. But in our conversations with patient advocates and disease foundations and patients and oncologists who focused on late stage patients, they said, hey, if you had a CLIA certified test, we would just try this. And we appreciate you don't have much clinical validation data, but we would love to see how our patient cells respond in your hands against a wide variety of cancer drugs. So we went down the CLIA certification path. And here's sort of the workflow to give you an idea of what this looks like in a real CLIA certified laboratory. So what happens is we do a biopsy, and we're CLIA certified for all the fluids and all the tissues. So we get a biopsy done. We have a little shipping kit. It's a little FedEx kit with tubes in it. It's a couple of 10 mil green top tubes for solid tumors and 3 50 mil tubes for the liquids, for the pleural effusions and ascites, with a drop of heparin at the bottom. So you have to talk the interventional radiologist out of putting them in formalin, one of the key challenges in our lives. But what we do is we get who's ever drawing the cells to squirt some of those cells into one of our tubes, put it in our little shipping kit, press the temperature control button, which reduces the temperature of the kit to 4 degrees C, seal up the kit, and walk it down the hall to Federal Express. And we say to people, if the day you do the biopsy, you got to get that thing out to us. If it's the next day, chances are the test won't work. Those cells are going to be dead by the time we get to them. But you got to ship them same day as you biopsy them. So those cells are shipped to us overnight. We take them out of the box, and then we plate them. Plate a bunch of wells for drug testing, a bunch of wells for controls. And I should say, prior to plating them, we do a purification, okay? Because we need a high concentration of cancer cells to make the measurement and the statistics downstream work. And I'm not going to do a lot of slides on purification, but to give you the general idea, in liquid tumors, it tends to be much easier. So for myeloma, they tend to be CD138 positive, and you just do a bead-based purification and pluck out the cancer cells. That works really well. For the solid tumors, no such luck. There's no such surface marker that we can use beads to purify. So instead, we do a negative selection. So lyse all the red blood cells, lyse the T cells, lyse the stromal cells, and reduce it to what we hope is a highly purified set of cancer cells. We then run it through flow cytometry to measure how many of the cells are EpCAM positive, and we have a CLIA threshold of EpCAM positivity. If it's high enough, we say, aha, this sample is pure enough for us to run our test on it. So that's purification number one, kind of a biological purification. Once we have those purifications, then plate the cells, drug them, incubate them overnight, 24 hours. Next day, take all of those cells and run them through the SMR, to actually measure the weights of all those individual cells. But we have a second kind of level of purification at that point, which is imaging based. So we actually take a bright field image of every cell, and I really should say every particle going into the SMR. And then Rob and team have manually curated about 20,000 images of cancer cells. So if a thing going into the SMR is a clump, or maybe a doublet, or if it's debris and isn't spherical like in a cell, then there's a machine learning system, a CNN, that automatically recognizes that, kicks it out of the data set. So it purifies the data set one step further. We then take all of the measurements, create those weight distributions, statistically compare the distributions, and write them out into a very simple report for the 20 drugs that we tested, and send that back to the oncologist. This has two really nice clinical characteristics that our oncologists and patients really appreciate. The first is speed. The biopsy happens on Monday. The oncologist is holding that report on Wednesday. And for a lot of these late stage cancer patients, they don't have much time. We've talked to a lot of oncologists that say they don't even bother to send their late stage patients out for sequencing because it's a 2-week turnaround time. And they have to make a decision a lot faster than 2 weeks for these late stage patients. So the 2-day turnaround time is clinically very attractive. And the second thing is size. we can't do this from CTCs. We regret deeply, but we don't need, a core needle or a surgical resection for every one of these samples. We need about 5,000 cancer cells per drug that we test. So we can do a panel of 20 drugs with about 100,000 cancer cells. And we routinely get those in fluids, and we pretty routinely get those in core needles biopsies, but we on occasion can get them in a fine needle aspirate, a number 22 needle, punched into the periphery of the tumor where it's hot, if the doc knows what they're doing, they don't get us a lot of necrotic tissue, they get us a lot of live tumor cells. That's a very attractive assay for a patient because a number 22 needle biopsy is a very safe procedure. And here's just a picture, kind of a sight chart, sorry for the sizes here, but of what goes back to the oncologist. And we send back, it's a multi-page report and there's a bunch of QC stuff at the end, but the first 2 pages are the parts that really matter. So page 1 is just the list of the 20 drugs scored on this dimensionless metric 0 to 100, where 100 corresponds to high likelihood of response and 0 corresponds to low likelihood of response. And that's actually just a p-value mapped to a 0 to 100 score. For a lot of our oncology partners, they just look at this, they look at the top drugs, they look at the NCCN guidelines, they look at what they were thinking of doing, and they proceed. They never look at anything else. For other of our oncology partners, and actually some of our more educated and activist patients, they're very interested in the second page of the report. And what this shows that the vertical axis is actually exactly the same as page 1. And the scores on the left axis of the vertical axis are 0 to 100. So it's exactly the same data. But the horizontal axis is really interesting because this is our actual mass measurement. It's expressed in 2 ways. One is in picograms and the other is in percent of mass change. What percent of the mass changed when we applied each of these drugs for 24 hours? And it gives you kind of a nice feeling for how the drugs are responding. And another way to kind of interpret this chart is the horizontal axis, mass change, that's signal. That's signal, but it doesn't speak to what's going on in the measurement system, which has a lot of noise in it. So the vertical axis is a measure, a statistical measure of signal to noise. It's what is the probability that this signal is not a random fluctuation in this noisy biological system? What's the probability that this signal is indeed a real mass response of this drug against those cells? And we have a very kind of sophisticated and formal statistical approach to doing that calculation, which generates us a p-value, which we can then map to the scores. So this is the information that the oncologists are getting back. And the last thing I want to say, and leave it open for we'll have 15 or 20 minutes for questions, is kind of where do we go from here? I mean, our primary focus is patient testing. We've done 50 and we'll do another 100 plus this year and validate if this novel mass biomarker really can predict which drugs work for which patients. But there are other things to do with this kind of incredible measurement tool. One of the things we will have, and we won't have it this year in 2023, but we'll have in the next few years, is a big data set. And I just kind of speculate here. What if you had 100 drugs? We're not testing 100 for each patient. We're testing a subset of like 20 of those 100. But you had it for 1,000 patients, and you had this 100 by 1,000 matrix with about 20,000 data points in it. That's a really interesting data set. As I mentioned before, this ends up being kind of a strong proxy for actual patient outcomes. And then you start learning things about on-label drugs. For example, if you keep running the NCCN drugs over and over again for patients with the NCCN indications, you may find that the frontline drugs don't work quite as well as the second or third line drugs. That would be really interesting information for a drug development firm that owned a third line drug, right? Because they could potentially elevate it and have a lot more use of their drug. Similarly, the thing that we are particularly interested in is running off-label drugs. The whole NCCN on-label approval process is very partial. We haven't tested every drug against every cancer. And the way we make decisions about what's approved and not approved, if the experiment's never been run, then it's not approved. It doesn't say it isn't going to work. And it doesn't say it's not going to work for a few % of the patients who would get it. It just says it's not approved in the NCCN guidelines. So our vision in the future is running a boatload of off-label drugs and building up enough data, we can go to the owner of the drug and say, hey, you don't want to spend, we appreciate you didn't want to spend the $100 million as a shot in the dark for taking your drug and applying it to a new cancer. But we've just run it over hundreds of patients, and it seems to work off-label really well for these indications. that could expand the usage of drugs across cancers and across patients. And kind of at a public policy level, we really hope we make this change. I mean, think about what are the drugs that we have that are approved, that are FDA approved. It's, I counted them last year, it was 273. It's more now. Around 300 FDA approved drugs, and the amount of money that's gone into approving those drugs, again, if you kind of count up the R&D budgets, it looks like to me about $1.5 trillion over the last 50 years. And yet the clinical trials methodology for determining exactly which of these 300 drugs work for exactly which of these 61 cancers, now becoming hundreds of cancers as we begin to understand subtypes, is horrifically flawed. We just don't know which drugs work for which cancers and which cancer patients. So if we can shed light on which drugs actually work for which cancer patients, essentially we're leveraging this $1.5 trillion asset without having to do more clinical trials. And that's a really valuable thing we hope to do. And then some other cool things in the future are sort of correlating this mass measurement with additional measurements. We're doing some of that ourselves in-house. We take a bright field image of every cell. We do no sophisticated image analysis right now of those cells. And if your lab is doing sophisticated image analysis and would like to partner with us, we'd be thrilled, because we think we have a huge amount of information sitting in those bright field images that we don't know how to take advantage of. We also, it's pretty easy to get volume information of a cell. And if you have mass and you have volume, you can calculate density. There's a literature that says, cells are really good at maintaining their density. When that's perturbed, it's meaningful. Again, a really interesting research subject that we haven't done much on, but it's a fruitful thing to do to kind of fully exploit the power of this mass measurement technology. And then external measurements, correlating this with genomic measurements and proteomic measurements. Again, we've done none of that at all. We're just too young and too small to have done it. So we think we've just scratched the surface of this kind of amazing mass-based biomarker, a biophysical biomarker that has this wonderful universal quality. What's the common factor that all cancer drugs do? They affect the growth of cancer cells. And so that's a universal quality, and has this wonderful integrative property. This biophysical mass measurement integrates all the details of molecular mechanism, of genomics and proteomics and metabolomics and epigenomics and all those sort of extremely complicated molecular phenomena, integrating it together at sort of a macro phenomenon, a Liopoint of function for all you mathematicians out there who to enable us to make an important clinical statement in the absence of perfect information around the molecular mechanisms of action. So with that, I want to stop. I'm definitely going to bring Rob into the Q&A. The commercialization questions I'm happy to take, but any of the detailed technical questions I'll direct to Rob. So Tony or Keith, back to you guys.
Yeah, thanks so much, Cliff. So we're definitely open for questions. I think we've got one from Tony. I don't know if Tony, you wanted to just.
Sure. Thanks, Cliff. That was great. Early on, you mentioned that you have 3 active clinical trials, and I'm wondering, I can imagine a couple ways you'd have these. One is that you are simply following patients who are being treated according to dealer's choice and seeing how well you predict response. Another would be that you're actually using your assay to assign therapy. And could you tell me which of these you are running there?
The former. We do no interventional trials right now. We're not far enough along to do that. But so these are all observational trials. But as I mentioned, they're prospective observational trials, because we actually test a broad range of drugs. We have predictions across as many as 20 drugs. And then invariably the oncologist gives the patient one or maybe a combo of 2 of them. And so we have that data set, even though we didn't direct that oncologist to give them the drug, the oncologist picked the drug. But it absolutely, where your question I think is going is that you can't calculate how, a better patient outcome associated with that, because we're not directing the oncologist away from that drug. And that's a true statement. And that's something, when we go for CMS reimbursement, we're going to have an interventional study that actually changes the therapeutic decision. And we plan to do that. We're just not there yet.
Right. And this is just a note, I would hopefully someone will be figuring this out for our field a little bit, but something for any company to consider is, I don't know the impact yet.
Yeah.
So you'll have to go through the FDA at some point, probably through an investigational device exemption. And you're probably aware that there have been some regulatory changes, including their willingness to consider whole platforms. And they're a little vague on what they mean here.
Yeah.
But you can imagine that the regulatory concerns would be much greater if you're going to have to, you and other companies like you are going to need to submit to regulation for every single drug disease combination, as opposed to maybe a system that would be better for us, where they would say, oh, we've seen some demonstration of what you do for any panel. You don't need to justify this for every single drug and every single tumor type. You have a sort of panel that works, just like you don't need to justify sequencing for every different nucleotide and every different tumor that you study. Have you guys thought about that?
Yeah, we have, and it's a well articulated question with all the complexity built right in there. We actually engage McDermott Plus, which is one of the premier CMS FDA regulatory law firms in Washington, D.C., to sort of help us ask and answer those kinds of questions. And the answer is very different for FDA and CMS. FDA, as you note correctly, has begun to be more open-minded about these platform-oriented approvals. And I think that, the NCI Match trial and iSpy and things like that have sort of guided them in that direction. CMS, not so much. And so our thinking when we go for CMS approval in this is we're going to probably have to do a breast study and probably have to do a lung study. And then hopefully after that, we can talk them into a carcinoma study. But initially, we think it'll be tissue of origin. So it's the additional bad news is it's a moving target. These regulatory processes and criteria tend to change year to year. It's also a moving target because in the CMS world, they're nine max, 9 of these contractors, and they all use different standards by which they approve or disapprove assays. So very complicated reimbursement environment. And we really just started the process of figuring out how to get there.
And I hope the SFPM will help. We'll hear more from Keith someday about this. I think he's forming a working group to try to help be a nucleus of having more of a unified voice in addressing these from an FPM perspective.
Yeah, I hope so too. And maybe doing some innovative, trial design for the community of functional precision medicine would be really, really helpful. For example, we had one interesting conversation with Optum Health, and they're beginning to think a little bit creatively about getting, diagnostic tests into their patient population, and they of course are insuring that population. They make sort of a throwaway comment, hey, when you're running a trial, we are the control arm. For any cancer you pick and any drug you pick, we have 1,000 patients who've already had that drug and with that cancer. Why would you run a control arm in your study? So there's a synthetic control arm capability already built in, but I think we need to take a real leadership role for FDA and CMS and maybe in partnership with a group like Optum Health to show them that this doesn't conform to their standard sort of RCT model, but it actually, meets the demands of rigorous data analysis.
Yeah, and just to emphasize, like Tony said, we're convening that work group now. And so if people on the call would like to be involved or interested in regulatory issues and helping to solve those, definitely reach out to myself or Tony or anyone, and also refer others to us if they're interested. It can be people from different groups and different perspectives will be really helpful as well. One thing, Cliff, with such a systematic chip-based technology and the speed with which you can do things, seems like you have an opportunity for combination therapy, which is something that many people in the field of oncology has really been blocked at trying to query combinations, both in the research space as well as obviously in the clinical space. Maybe tell us a little bit whether you've thought about that and how you might approach that with this technique.
Yeah, we're in the early stages of figuring that out. We haven't run any experiments yet, but I mentioned the method that we use to calculate the response of cancer cells to cancer drugs potentially lends itself to doing a calculation, sort of an in silico drug combination. And so we are kind of hot on the trail of that. We have a very accomplished statistician who's working on that. It's wildly complicated statistical process. My PhD is not in biochemistry. My PhD is in mathematics and in complex systems. I was once a statistician, and this is frankly beyond me. So it's a very complicated piece of statistical work. But I think sometime in 2023, we will indeed be putting forward kind of hypotheses about what we might be able to say about drug combinations, based on testing monotherapies and then computing the potential effect of drug combos on patients. But too early for us to report on any results there.
Yeah, I think Ken Dixon, you might have a question. Is that right?
I can read. Ken Dixon sent me a question, which is that do you have the same 20 drugs for every tumor, or do your drug panels change for each tumor type?
We have a different drug panel for each tumor type. If you actually go on our website, the 39, or I think maybe it's 35, you check out the leukemias, the 39 drug panels are there. And we have basically an algorithm for creating a drug panel. The first drug we run for each of the 39 NCCN guideline drugs are the on-label drugs. And we find that, this is what our physician partners want first and foremost, because these are reimbursed. And without reimbursement, it's very hard for them to give a drug to a patient. So now we haven't validated all 273 of the drugs. We've only done 100 of them. So we haven't, there's 69 FDA approved anti-cancer drugs for breast cancer. And we've done, 16 of the 69, and we're started working our way through the rest of them. So the first part of our panel is always the NCCN approved drugs, and it's TKIs, inhibitors, and then sometimes, other things in there, other interesting molecules. If we fill out the NCCN part of the panel and we still have room left in the 20, then what we do is we drop in some inhibitors that, based on a database that we have access to, are simply the inhibitors that hit the most targets. Because what we're looking for is off-target activity. We try the inhibitors that hit the most targets. And so we're kind of systematically running a set of inhibitors as part of the panel of 20. In addition, when we find physicians, when we're working with physician and their patient, often the physician will say, my patient just failed the first 2 on your list. We'll drop those out and add 2 more in because there's sort of no point in running that test. On the other hand, if they say, my patient failed those 2 inhibitors 2 years ago, we say, let's try them again, because we see resensitivity in patients. And without some scientific evidence, you probably wouldn't put the patient back on a drug they failed 2 years ago. But we've seen in many circumstances that if patients are resensitized to drugs that they failed a while ago. So yes, we start with the NCCN guidelines, we fill it out with other drugs, but then we work one-on-one with physicians to make sure that the panel makes sense for their patient.
Thank you very much.
Well, do anyone else have questions at this point? If not, then I think we'll go ahead and end the discussion. And thanks very much, Cliff, and the team at Travera. Really appreciate you sharing your work with us.
Great. Thank you very much, all. If you have any questions, please reach out to us. You see the email addresses there. All right. Thank you all.
Take care, everybody.